animal virus updated PowerPoint for coronaviruses. This is our third, excuse me, video audio. The last one was on virus attachment or absorption to host cells. So we're kind of walking through the stages of human animal virus replication. So the first step is attachment or absorption. The second step is entry. So this is step 2, and that's what this short, hopefully short video will be about. So there are 3 different ways that human animal viruses can get their nucleic acid into the host cell. And the 3 different ways are um, direct penetration, which is really similar to what bacteriophage do in injecting the viral nucleic acid into the cell, or there's fusion. This would occur with envelope viruses where the envelope fuses with the host cell membrane. And the last one is kind of the Trojan horse approach called endocytosis, where the virus tricks the host cell into bringing the virus inside the cell through the process called endocytosis. So we'll just go through these really quickly, guys. So just to get you oriented, this represents the surface of the host cell, and these little pink plates would be the host cell surface receptors. And here is our virus. Now my understanding is that usually this direct penetration where the virus just inserts the viral genetic information of the cell, my understanding is this is usually with naked viruses, but I'd have to double check on that. But one example is polio viruses. Okay, polio viruses are RNA, naked RNA viruses. So the virus would attach using its adhesins and binding to specific cell surface receptors. And then proteins within the capsid would create a pore, a channel, through the host cell membrane. And it's through that pore or channel then that the viral genetic information would be injected into the host cell. So that looks a lot like bacteriophage entry when the bacteriophage inject their DNA into their host cell. These next 2 are really different from bacteriophage, right? And so this next one called membrane fusion, this is only going to happen with envelope viruses. And one article I was reading said maybe coronaviruses use this membrane fusion, but the medical microbe book I read said they thought coronaviruses did this endocytosis. But anyway, so we'll put maybe coronaviruses here. Okay, so you guys, let's pretend this is, oh, I know for sure, you guys, let's use HIV. Okay, so HIV, it's an RNA virus, it's an envelope virus. So this could be our HIV. This is the envelope, and the HIV adhesins in the envelope are called GP120, right? So HIV GP120 will bind to the host cell surface receptor, and the only one I'll mention, you guys, is called the CD4 molecule. Okay, so these would be the CD4 molecules. So HIV binds, and then what will happen is another viral protein will trigger fusion of the viral envelope with the cell membrane. Now remember, the viral envelope is just stolen host cell membrane, right? Fluid mosaic, consistency of olive oil. So with HIV, the fusion protein is called GP41. It triggers fusion of the envelope with the cell membrane. Okay, so there's a fusion, and that permits entry of the capsid. And then the capsid proteins will be disassembled, releasing the viral genetic information, and then that poor cell's in trouble. Okay, so that's called membrane fusion, and we would see it in some envelope viruses. Now this last technique, endocytosis, I think of this as the Trojan horse approach, where the virus is like knocking at the gates of the cell saying, hey, I've got a great present for you, bring us inside your cell. So again, folks, in the medical microbe book I read, it stated that endocytosis was how coronavirus, like the pandemic coronavirus, would get into our cells. Influenza viruses also use this approach to get into our cells, and later we'll talk about herpes viruses. They also use this approach. So here we have, in this case, if this is the coronavirus or influenza virus or herpes viruses, this is the envelope with their receptors. Okay, they bind to the receptors on the host cell. And again, you guys, they kind of trick the host cell into thinking, oh, this is something good. And so the host cell, it endocytosis. It has this invagination of the cell membrane right here. So this is a process of endocytosis and brings the virus inside the cell in this endosome, this membrane bound vesicle. And remember, you guys, the envelope virus is inside. And then at that point, within the virus envelope, there'll be a fusion protein, a protein that again is going to trigger fusion of the viral envelope with the membrane of the endosome. And then we get release of the capsid with the viral genetic information into the cytoplasm. The capsid, the capsomeres are disassembled, and that releases the viral genetic information. And again, that little cell's in trouble. So the three ways that viruses get into human cells or animal cells: direct penetration, fusion, or endocytosis. So folks, we'll end this video here, and the reason is this next set of slides, human animal virus replication synthesis, so synthesis of the viral genetic information, synthesis of viral mRNA, synthesis of viral proteins. This gets really thick as molasses, thick as fudge. So we'll stop the penetration audio here, and we'll have the next audio will be on synthesis of viral nucleic acids and proteins.